Hello, everyone. Thank you all for joining me today. I'm going to be going over just kind of an array of projects. So the name kind of tried to fit all of those. So first, we're going to talk about ESI-MS of aqueous polymers with rapid, simple deconvolution techniques, or a single one. And then I'll go over a top-down proteomics technique that we've been working on in our lab using polystyrene sulfonate oligomers as noncovalent selective adduct protein probes. And then the remainder of this talk, I will talk about different mass spec approaches that have been used to attempt to understand the formation of synthetic melanin. So yeah, to start off, as I said, and please feel free to either interrupt me as I'm going, or I was going to just stop after the end of each part and open it up for questions if anybody wanted to chime in at that point too. So the first part is talking about measuring homopolymer size from ESI-QTOF spectra using a UniDec algorithm that was created by Michael Marty from University of Arizona, which I will go over in a second. So problems with traditional ESI and QTOF analysis that we've seen of polymers is include multiply charged ions that can exhibit a low mass bias. We can see in the example of polyalanine here. Right, let me put a laser on. That we get, it's a molecular weight range of 1 to 5 kilodalton, but we get ranging from 100 to maximum about 2,500. And then the error exponentially increases for larger polymers. We can see if we try to run a 7,000 kilodalton polyethylene oxide, we only observe it from about 1 to 2,000, and it's extremely noisy and complex. We also have overlapping distributions that can limit any sort of N-group determination. And so for this, multi-TOFs have been considered the gold standard for mass spec polymer analysis. However, other online separations such as SEC and charge deconvolution techniques have also been reported in the literature. And so those are going to be, or today I'm going to talk about a type of charge deconvolution for polymer spectra. So charge deconvolution simply is just decoupling the charge from the overall mass spectrum so that you obtain a zero charge mass spectrum. So an example of myoglobin, the protein, being deconvoluted is shown here. So something like myoglobin can exist in a lot of different charge states, and so that creates this dispersity that then convolutes the analysis. And so ultimately, using charge deconvolution techniques, you can obtain a spectrum that yields just the exact mass rather than a mass to charge ratio. So these are done through either peak assignment or isotopic methods for simple techniques. So peak assignment being just picking out the peaks and then assigning the charge state based on, taking the mass and then dividing by expected charges and then assigning peaks that you observe based on the expected M over Z. Isotopic would just be the opposite. So looking at your spectrum, taking the isotopic resolution or the change in M over Z, determining the charge state, and then back calculating your mass of your sample. However, you need a lot of simple techniques or simple spectra to do something like that. And so the simulation-based methods have been more useful for complex mixtures, and they also are considered quantitative because they undergo a series of statistical analyses that fit either a minimum chi or maximum entropy type data sets. And so it's been shown in the literature on polymer spectra from various groups. And so we can see that charge deconvolution was capable of distinguishing polymer samples of various, polymer distributions of various oligomer lengths and adduct ions. And then more recently, a paper came out showing this. Sorry, let me hide this. There we go. Oops, sorry, guys. It's just stuck on here. Okay. More recently, a paper came out showing this kind of emerald city pattern that I think the McCluskey group published. And so they were able to show or distinguish and deconvolute isobaric isomers of dextrin that exist at a variety of different oligomers, and then the charge states of those different oligomers can result in overlapping isobaric ions. So these techniques are a little complicated and require a lot of statistical analysis, computational programs, or software licenses that can be expensive. And so, as I mentioned, the Michael Marty and Carol Robinson in 2015 developed this Bayesian deconvolution technique that they call UniDec, or universal deconvolution. And it's supposedly a quantitative technique that uses a probability-based outcome using Bayesian statistics. And so an example of what this shows is that you would input your mass to charge spectrum that you obtain, so just the text file. It will then iterate for all possible charge states based on an inputted range that you tell it. And then from there, it will fit that data and determine what the probability of that charge state and that peak being present for a given M over Z. So the example I'm showing is the example data from software of phosphatidylcholine nanodiscs that have a various degree of phosphorylation, I believe. And so from that deconvolution, you can then get a mass to charge kind of heat map that shows you the average charge state for each mass to charge ratio or peak that was observed. And so from there, that can then easily, that's basically this spectrum right here, or plot, that can easily then be transformed into a single mass spectrum, or single mass to charge 2D heat map. And so now at this point, this plot is showing the intensity of a given mass and then the average charge state that that was observed at. So then they can ultimately develop an intensity against just the actual mass of the species. So they were looking at things over 100 kilodalton and have been able to show very promising results. This has been used on direct injection, ESI, LCMS, ion mobility, MS, more recently charge detection papers, as well as just high throughput processing. So it's a very versatile software, easy to use. It's on GitHub for free. So the objective of what we were doing with polymers is just to analyze 5 diverse polymer standards and incorporate this technique into, or to see how well it can interpret molecular weights and the number average of polymer. And so for here, I'm looking at the wide polypeptide standard of polyalanine, so 1 to 5,000 daltons, and then polyethylene oxide ranging from 1 to 7K, so 2 different standards, a low and a high mass, and then as well as a polystyrene sulfonate that's sold as 1 kilodalton, but from 2 different manufacturers, so it differs by about 500 or a couple of monomers. So I felt like these were 5 good, just diverse standards to utilize the application on homopolymers. So the first analysis is of the poly-D-L-alanine, and the average molecular weight, as I said, is from 1 to 5 kilodalton. Oh, sorry, it should just be dalton, not kilodalton. Whoops. And so here we have the raw data or text file that's inputted into the spectrum, and we can see that it then finds all of the charge states for those given mass to charge peaks. So we have a good amount of doubly charged ions kind of fading into the triply charged, but that ultimately is shifting our distribution to the low mass region. And so then you can, using the software, it then deconvolutes that to give us the mass and the various charge, ultimately yielding a 0 charge mass spectrum that shows us exactly what it's reported to be as between 1 to 5,000 dalton polymer, and it actually appears to be 2 separate distributions, one at around 4 and then the other at around 1.5 to 2,000. So that was interesting, exciting results for a broad standard, especially because mass spec has trouble quantifying broad polymer masses, especially in MALDI if the PDI is too big. So then looking at,
I'm sorry, I do have a question here. Like, if I got it properly, this tool is based on statistics or probability, right? Like the base. So does the program also give you like a level of confidence in the assignment?
Yeah, it gives you they call it a uni score or a D score, and so those each peak that it fits the corresponding score with it, and that's on a 0 to 100 scale. So I believe they're in the D score and the UniDec score or Uniscore are inverse, I'm pretty sure, but they're basically confidence or probability.
Okay,
score.
so then your inputs are the range of chart set that you expect from 1 to whatever you want maximum, plus a minimum level of confidence for the peak to be say, okay, that's the correct chart state.
Yep, exactly.
Okay.
Yeah. So I can, there's a bunch of different settings that you can go through. You tell it the expected charge, and I usually overestimate. You tell it the observed mass to charge ratio of the spectrum you're inputting, and then you also tell it the expected molecular weight of the species. So it was designed for biopolymers, so much more monodisperse species, but like I said, I usually overestimate and then narrow in on the species. So here we have the, yeah, for sure. Here we have the 1 kilodalton polyethylene oxide standard. So we can see our raw data was then fit, and it overlines nicely here. You can see each peak corresponds pretty much to this intensity plot, and then showing you what charges they exist at. So then the zero mass spectrum yields a distribution that's ranging from the expected range, and then like Theory just asked, it does give a confidence score, and so each peak here can be extracted as a mass and intensity text file, which then I went into Excel and calculated the number average molecular weight, and was able to get a MN of 900. And then for the 7 kilodalton polyethylene oxide, this one was more exciting because of how good it looks for what we typically can see on electrospray. So here we have the raw data, looks not much different from the 1 kilodalton, as we can see how much bias we have in the low mass region. However, if we look at the average charge states, they're ranging from 5 to about 16. And then ultimately that generated a mass spectrum, zero spectrum in the approximately 8,000 range. And the calculated number average molecular weight was around 8,000 as well from that data. So overall, of the polymers that I tested, like I said, I looked at polystyrene, 2 different polystyrene sulfonates and the 2 PEGs, or polyethylene oxide, either one, and was able to calculate both the raw average number average molecular weight and as well as the deconvoluted, and then I compared the errors relative to the reported mass of the standard. So all of the polymer standards that I used for GPC standards, or the PEOs for GPC standards that were reported in MWs, and then the PSS for PSS, the larger one was reported as an MW, and then so was the smaller one, but the smaller one was also reported giving an MP, which they called the maximum, or the mass at peak height, or maximum peak height. So that was a significantly lower mass than their reported MW, which is, I'm pointing out because the average error in our deconvoluted spectrum then is in much better agreement with that MP value than the MW value for that polymer. But overall, we have pretty good agreement, especially relative to the raw spectrum. As we increase mass, we see it's an 87% error with the 7K, and down to just a minus 10% error once deconvoluted. If you look at that in terms of the just degree of polymerization or average oligomer count or NMR, you can see that for the expected N or average N value for PSS, the 2 different ones, they were expected to be differ by about 3 oligomers, and the raw spectrum showed that they only differ by about 2. However, the deconvoluted was a little bit of an estimate, but it did show the greater degree of difference between those 2 polymers to begin with. And then again, the largest difference or importance was shown with the 7K standard, and we had a percent difference of only about 4.7 when compared to the expected count or oligomer count. So overall, just some conclusions from that. It was pretty much, required a high resolution instrument. So we used ESI-QTOF on our SNAPP, and then the UniDec showed that it was capable of performing quantitative studies in good agreement with the accepted standards. The oligomeric resolution was actually possible with increased sampling on the 7K PEO standard. So if you just tell it to look, or if you tell it to iterate for a shorter distance of mass points in the software, it is able to then find those different oligomers, so you can do the same kind of mass spec analysis that you would with something like MALDI. And then it's suitable for both broad and narrow standards. So the advantages are obviously that it's a free, open source, user-friendly software. It's a greener technique than GPC and SEC because it doesn't require calibration. You can directly analyze the species and get a decent mass analysis that way without consuming a ton of solvent. Limitations of this are that it does glitch a little bit, the software, if you overload it with too much data at once, and the increased accuracy in your analysis obviously comes from increased computing power, but everything was done on a just typical computer. This isn't supercomputing or anything that's special. The i7 processor. And so future work with this that I'm interested in is looking at the abilities for this to distinguish copolymers or heterogeneous mixtures. There's already a mass defect application built into the software for proteins, so it could be interesting for polymers as well. And then be interested in comparing the results to obtained on either trap instruments or higher mass quadrupoles, and just see how you could compare data sets from instrument to instrument. So that pretty much summarizes that part of the talk. If anybody has any questions on how it works, I'd be happy to go over some of that more. I'd also be happy to send you any papers or anything that you'd be interested in.
How high would you expect them to go in terms of molecular weight? Like, I mean, what would be the limit of your ESI-QTOF system, like to detect a polymer? I mean, to have at least a decent signal for the multiple charge species.
Yeah. So it's definitely, we're limited by our 4K quadrupole. So I think I tried a 20 and that was not very good of an analysis. So we're pretty much limited by, I would say, 10K on our instrument. However, as I showed earlier, the UniDec example data was done on Carol Robinson's, I think she has a 32K QTOF. So they were capable of seeing over 120,000 molecular weight samples that way.
So then in your case, it's because the polymer doesn't get enough charge to multiple charge enough?
Yeah, and then just the
yeah.
quadrupole can't transmit those larger species based on the applied voltages that it's giving or potentials. So that's pretty much the limit there. But yeah.
I know Brian Katzenmeyer's on the line right now. I'd be interested in seeing if he'd be willing to work with you on he's got a MALDI MRT, but I believe he also has electrospray capabilities on there. So now you can really extend that mass range out, and you probably get really nice high resolution.
Yeah, I was actually interested in reaching out.
Yeah,
to
thanks,
Brian
Tony.
Oh yeah,
Thanks
I
for the
saw
plug.
some beautiful data from him, so that's what I'm saying. I think this would be perfect.
Yeah, thanks for the plug, Tony. Yeah,
No
we
problem.
have with the MRT, which is multi-reflectron time of flight, that allows us to go up to 200,000 resolving power, excess of that for MALDI and electrospray data. But from a mass range standpoint, that does limit to about 9,600 to do that many passes in the time of flight region where this analyzer is at. There is an option to run in what we call a diamond mode, which does decrease the mass resolving power, but it extends the mass range up to about 20K. And that's pretty good for those, obviously on this call, you're familiar that in an orthogonal accelerated time of flight mass spectrometer, there are limitations, because moving those polydispersed ions around. So that's why linear TOFs, like the multi-TOFs, as Addie correctly pointed out, have some advantages to that type of setup. But there is some options there as well. And I was actually, Addie, I was thinking about, you mentioned about some of the limitations or advantages of this technique that you're using with the software. I mean, you mentioned about GPC, or SEC, as using lots of solvent, but, there are fast columns available, or there's even systems available that can do the analysis within several minutes, consuming very little solvent, only a few mLs of solvent potentially. So I'm just curious to see what, as an industrial lab, if like Tony or Theory or anyone else on today's call has, would be wanting to do this by MS versus just traditionally just doing this by size exclusion chromatography and getting the result done from an MN, and and getting all your molecular weight distribution data that way.
Yeah, we typically do GPC for that higher molecular weight polymer. Something that was low molecular weight, which might be biased by GPC, we would get by mass spec, but
Right.
and from an industrial perspective, that's how we're focused. And
Yeah.
and a lot of times our higher molecular weight polymers tend to also be copolymers and things like that, which are even more difficult to do by mass spec.
By mass spec. Yeah, thank you, Drew. Yeah, I was thinking along the same line, same thing. So I was just kind of curious to see if anyone else feel that same way. Just because I know solvents can be expensive, but with today's columns and in some cases some systems that are available by the various vendors, you can definitely take advantage of that and run your analysis in less than 10 minutes is a typical runtime nowadays. So solvent consumption is not as bad. And you can also get very accurate molecular weight information if you're using detectors like a light scattering or a viscometry and those types of options if people want to get more detailed than just say refractive index.
Yeah, I definitely agree. I think it's something that could be cool to couple with your LC or SEC elution. So just doing an online separation, and then it could be a way to validate whether the hydrodynamic radii is affecting the analysis too, just because of you could then get the mass as opposed to the size of the species that's eluting. So could be something to validate. I was also thinking it could be interesting for synthetic labs that maybe don't have their GPCs running and calibrated every day. And so it would take them a full day to set it up, but this could be a rapid way for them to just analyze their material and keep going.
Yeah. And assuming they have a mass spec that can do it too.
Yeah.
Yeah. Not every lab does. So
yeah, no, definitely.
But
more than multi, but yeah, I get you.
Yeah.
Yeah, it's more likely that labs have a GPC than they do a mass high resolution mass spec.
Yep, exactly.
Yeah, just from cost. Yeah.
Yeah. Yeah. Thank you, Addie.
Yeah, no, definitely all good points. If no one has questions then on that, I'll move on to the next project. So this is a little bit more of a biochemistry project that is applying polyelectrolytes to protein footprinting. So using multidimensional mass spectrometry techniques. So for those of you, sorry, I don't know, my taskbar keeps freezing on there. For those of you unfamiliar with proteins in general, I know a lot of you are more material science people, the little animation on how proteins fold. So the folding is driven mostly by the hydrophobic effect and expelling water from the system, but ultimately proteins fold by forming their peptide bonds through secondary structures, and then forming intermolecular and intramolecular hydrogen bonding with those nearby residues to create these alpha helices and beta sheets that then ultimately fold into these tertiary structures. And so we're interested, or protein footprinting is the process of determining what residues are on the surface of that protein, because from a biochemistry standpoint, these are going to be the regions that might be the active sites of the protein. They might be sites you could inhibit for drug treatments or anything like that. It also tells you a lot about the pathways of the proteins themselves. So that's typically done using either bottom up or top down techniques. So bottom up being an enzymatic digestion of the protein that you then run LCMS or MSMS on the digested products themselves, and then ultimately piece back those fragment ions into the larger protein. Top down is when you take the protein and inject it into the mass spec directly or with LC to help purify. And then from there, you can fragment and kind of break it apart that way. And so there are top-down techniques that are used for protein footprinting, including covalent cross-linking, hydrogen deuterium exchange, and then the free peroxide or hydrogen radical peroxide, oxidative products that can form. So this is an example of that FPOP or HRPF, sorry, technique in which they irreversibly label the protein with hydroxyls. This is a bottom up, and then they digest it and then ultimately determine the sequences that were tagged to figure out what was surface accessible. And then Julian Ryan from UC Riverside developed a selective noncovalent adducting protein probe about 20 years ago, and they used crown ethers to form adducts with any residue on the protein. And so this gave them stoichiometric information about the amount of residues that were on the surface that carried basic charge or basic functional groups. However, there wasn't any sequencing that was capable of this because that interaction was not strong enough to survive tandem mass spec. So we're looking at using ionic polymers or polyelectrolytes for top-down SNAPP footprinting. And so this has already been shown by Ben Chen in our group and this paper. And so they were able to form noncovalent interactions with polystyrene sulfonate, a polyanion. Sorry, let me go back. So basic residues are going to be any sort of residue on a protein that is amine functionalized. So these are your lysines, arginines, and sometimes histidines. So also symbolized as K, R, and H. So they can undergo a series of different ionization states. And at pH 7, they exist mainly in these regions. And so when they are charged, they can then interact electrostatically with anionic polymers such as polyacrylic acid or the acrylate form or polystyrene sulfonate in its conjugate base form. And so, as I said, this has already been shown from our group to form strong interactions with polypeptides. So a paper here is showing that polystyrene sulfonate and polylysines were able to form compact charged structures or compact structures that were more densely packed and stronger than the competing polyelectrolytes that were chosen. And then, as I said, this paper revealed that the tag was capable of surviving top-down proteomics or tandem mass spectrometry through collision-activated dissociation, ultimately able to identify the region of the adducting that's occurring. Our objectives right now are just to present and work on site-specific methods to identify surface-accessible basic residues on folded proteins that range from 2 to 20 kilodalton and isoelectric points ranging from 4 to 12. So isoelectric points are just the charge of the protein at a neutral pH. So something with an isoelectric point of 4 would be an acidic protein, 12 would be basic. And then we're also interested in incorporating molecular dynamic simulations to probe distances between two basic residues that form a salt bridge with a given polystyrene sulfonate probe based on the maximum distance between the sulfonate residues on n number of oligomers. Lastly, we're interested in studying the relative binding strengths of the residues with polystyrene sulfonate depending on the polarity of nearby residues. So the workflow that we do is we take the protein and incubate it with polystyrene sulfonate and ultimately isolate this complex species. This is done at under biological conditions. You might have to purify it if necessary, and then it's directly analyzed through ESI-MS using collisionally activated dissociation. And what happens is we're able to cut apart that protein, yielding fragments that are just naked BY fragment ions, and then what we call the B star or Y star fragments, and those are going to be the ones that retained the PSS tag. There we can say that the ones that contain the tag are surface accessible, and the ones that don't are likely not surface accessible, either because they're interacting in a dimerization or because they're actually on the inside of the protein. So here's an example of data obtained by one of our colleagues, Peter, and then analyzed. So when we analyzed it, we were able to see that the melatonin, a pretty basic protein, it's a honeybee venom protein, was complexed with polystyrene sulfonate tetramer right here. So we isolated the +4 ion and then subjected it to collisions. and we're able to see strong complex fragments starting at the Y13 and then all the way down to the singly charged Y6 star ion. And so that corresponded to the N terminus, so Y fragments being, or sorry, the C terminus are the fragments that are the Y ions. And so if we look at the sequence, we can see that that is a very basic region of the protein. So we have lots of sites that the polystyrene sulfonate can adduct with, as opposed to the earlier region of the protein, or the region towards the N terminus, that only has one basic residue present. And so from there, we can then look at an example of the known structure of melatonin. So if you look at it memorized in based on its PDB file, you can see that some of the residues are interacting with each other in this intermolecular dimerization, leaving only a couple of them surface accessible. And so if we look at that from a different angle, we can see where exactly the C terminus is. And then the first fragment we observe is at, for the biochemists or people who care, it's at the proline kink. So it's going to be at the first at the weakest site of the protein. And then we're able to retain that tag all the way down to the basic residues, giving a site-specific analysis of the protein itself. So here's that modeled then with a polystyrene sulfonate tetramer and showing the adducting regions that could potentially be occurring in this tag. And then we're also looking at modeling the polystyrene sulfonate using molecular dynamic simulations. So here we can get the average bond distance between adjacent sulfonate groups to ultimately see the distances that, or the maximum distances that they could form these like bidentate bridges, bridges between nearby residues. So I had my colleague look at ubiquitin, one of the proteins we studied, in a in its neutral or biological state. And so he simulated that with the PSS tetramer. And here we can see that the R74 and R72 regions of this protein are going to be about, on average, 3 to 8 angstroms apart, as the polystyrene sulfonate has 2 potential distances. So when it's in this kind of pi stacked form, you get sulfonates that are only about 6 angstroms apart, and then they can also extend out to about 12 up to around 15 here. And so this also shows that the R54 and K63 are likely not going to be interacting with a single tag because their distance is too far away from each other to be bound in such a way. So ultimately, the summary of what we've done so far is looked at melatonin, glucagon, ubiquitin, and the epidermal growth factor. So these are proteins ranging from 2 to 8 kilodalton, and then isoelectric points ranging from pretty basic PI of 4 down to acidic 4.8. And so the potential binding sites are indicated here. And what's important to highlight is that we were still able to form adducts with our protein and isolate potential residues that are binding, and based on the naked fragments we saw, indicate the residues that are likely not surface accessible. So that's kind of the summary of this project. If anybody has questions on that, I'd be happy to go over anything there.
I may have missed the reason why, but why do you use the PSS tetramer specifically? Why not 5 or 6 or 3?
Oh, yeah. So actually, I think it was EGF formed with a hexamer. These three, it was just about the ion, like the tag that we were able to see. So I'm interested in fractionating and getting the monodispersed forms of each of those, and then seeing how they bind to the protein, like how the trimer, tetramer, pentamer, and all of that would bind differently, because I think that would give us an idea of the residues that are further apart from one another.
Have you tried using something like paratoluene sulfonate, so you just have the monomer? Is there an advantage?
I guess I haven't tried that specifically. At that point, I think that would be similar to doing, I think people do phospholipid tags. So it would be just a similar monomeric species. You lose out on the ability to then probe any distance between them. And then it would be interesting to see if the monomer is weaker. Yeah, so if that second interaction actually is what strengthens that electrostatic interaction to survive the collision. So it would be worth looking into for sure.
Yeah, I was wondering in terms of maybe being able to count the number of residues by the number of adducts with a simple small molecule type. I don't know if there's any need for that or advantage to that.
Yeah, that could definitely, especially if by themselves they'll retain to the point that you could then get those site-specific regions. Absolutely. I forgot. So my colleague and I are actually working on modeling polystyrene sulfonate in different polylysine oligomers, and then studying using a modified energy resolve mass spec technique, studying that interaction between different sizes and the amount of basic residues that it's bound to. So I ultimately want to look at something like a polylysine or a lysine trimer that's then been swapped with a glycine in the middle to see if that is stronger, weaker, unaffected by the binding. And we're also interested in looking at this on larger proteins that fold and create different allosteric or orthostatic conformations based on active site bindings to show that it is specific to that or selective to those solution-based structures. No one has any other questions? The last thing I just want to talk about is a very complex, almost headache of a project that I've been dealing with and working on, and that is synthetic eumelanin material analysis. So I'm just going to talk about a bottom-up approach, but ultimately we've done both top-down and bottom-up kind of pseudo techniques to understanding both the structure and formation of eumelanin materials. So synthetic melanin nanoparticles were first discovered after eumelanin was discovered in underwater by having underwater adhesive capabilities in mussels. It's a structurally diverse class of macromolecules, potentially even a sixth class of biomolecules if understood well enough. And then it has a broad absorption band with high conjugation. Species are able to harvest radicals and exhibit chelating effects. However, the exact structure and mechanism of their formation is still heavily debated in the literature. So the nomenclature and structural classification of these synthetic melanin nanoparticles can be split up based on monomers. So they are derived or thought to be derived from catechol or catecholamine containing species, in addition to cysteine, dihydroxyphenylalanine or L-DOPA, as well as dihydroxynaphthalene. And so these species form different types of synthetic melanin, as has been described in this paper. So they've classified them as allomelanin, pheomelanin, eumelanin, neuro, and pyromelanin. Most of this classification has come more from pyrolysis GC and structural properties rather than chemical bonding. However, here's examples of different types of eumelanin. So DOPA being the L-DOPA monomer, DHI being the dihydroxyinindole monomer, DHIC is the dihydroxyinindole with the carboxylate group still on it. So it's just the cyclized L-DOPA into the inindole ring right there, pre-decarboxylation. And so those plus additional dopamine and cysteine species have been classified as types of different backbones of melanin. And so there have been studies that have looked at the overall relative elemental abundance of these species and shown the carbon, hydrogen, nitrogen, and sometimes sulfur containing species, and as well as minimal oxygen in some of these. The analytical changes are very wide for this or these materials. So they're insoluble in most solvents. DMSO is one of the few solvents that you can get partial solvation in. They have extremely poor ionization efficiency because they absorb radiation and thermal energy so well. They don't want to like desorb or ionize as a result. They absorb across an entire UV-vis range, and as a result could be used as a multi-matrix, and so then you get mixed interference from the species itself when you're trying to analyze it, and it's interacting with the UV in a way that other materials might not. It's shown to have paramagnetic properties, and the mechanistic pathways seem to depend heavily on the media of the material. And overall, the mass spec data in the literature is pretty irreproducible from paper to paper and structurally inconclusive overall. Very few people have done tandem mass spec on these species to begin with. So unknown. Overall, people think it forms from, or this is the biosynthetic pathway of eumelanin from the Raper-Mason scheme, starting with tyrosine that undergoes oxidation either from the tyrosine oxidase protein or some hydrogen peroxide or other oxidant. And then that, along with DOPA, can form this DQ into the DOPA chrome and then DHI oligomers, decarboxylate, and then pretty much everyone just writes dimer, trimer, tetramer for stuff like this. So they're not quite sure how they oligomerize. People have proposed oxidative oligomerization or polymerization pathways that undergo either radical or quinone initiated mechanisms that can then form these diverse structures that are cross-linked, linearly chained. You've got pi-pi interactions, entirely hydrogen-bound, non-covalent interactions, and so on. There's also been cross-linked oligomers that have been proposed from the amine-quinone reactions that could potentially happen between the species. And then other people have proposed, and this has been a more, this has gained more traction in recent years, the idea that you have these smaller oligomers that can then undergo pi-pi stacking that then undergo these further proto-stacked molecules into these large particles ranging from 100 to 200 nanometers. The applications of this material right now is pretty diverse in terms of biomaterials, energy harvesting materials, and so on, but I'm not going to go into that too much. We tried looking at different bottom-up techniques by essentially observing the auto-oxidation of dopamine and L-DOPA in water. So if you just sit them in solution in a day or two, they will start to form these black precipitates and then become entirely insoluble in the initial aqueous solution. So what I'm going to present is the kind of bottom-up analysis of L-DOPA using ESI ion mobility mass spec as well as tandem mass spec of aggregates that form from solution. So here's the positive electrosphere ionization mass spec of L-DOPA in a water doped with methanol for ionization. And we can see the associated monomer here, the dimer up here, as well as a series of larger oligomeric clusters or aggregates that form. If we extrapolate the ion mobility or isolate the different bands, we can see that we form, it's a little hard to see, but up to n equals 62 monomer units in these stable aggregates were resolved. And so, and then in addition, each aggregate undergoes a series of hydrogen sodium exchanges, which is what you see right here. So as a result of the Zwitter ionic capability of L-DOPA. The interesting thing I saw in the data, though, if I went through and looked at each of these oligomers, is that maximum amount or the average amount of sodiums for each oligomer was approximate, or the average amount of oligomers for each sodium was approximately 4 to 6 monomers. So we can see that as the number of sodiums increase, we see approximate amount of L-DOPA staying in a about 4 to 6 ratio. So kind of chelating or coordinating to the sodium in a way that also fits the protomolecule species that form these, we call them porphyrin-like structures. And so that was interesting, I guess, from a structural analysis. So what we did next was confirmed that they were non-covalent interactions, in fact, by isolating one of the tetramer species with a single sodium and then subjecting that to MS, and we just see neutral losses of the unmodified neutral L-DOPA, indicating a non-covalent interaction between these species. If we isolate the, if we look at the arrival times for those species, we can then see that, so 811 being the singly sodiated exists as two separate conformations, and the doubly sodiated and triply sodiated, each undergoing hydrogen exchanges to maintain their singly charged, favor this more compact, smaller structure, indicating a kind of stabilization or packing favorability from the increased sodium. And then I looked at increasing the collision energy on these aggregates, and so this being the doubly charged region, so I was ignoring that, and overlaid their arrival times for the different DOPA tetramers with sodium hydrogen exchanges, and we can see that at the transition from 6 to 11 relative volts, we get an increase in the smaller or more compact structure that ultimately favors the same structure that the higher sodium or sodiated species favored, which indicated indicated that the collision energy increased increased the tendency for the smaller configuration, or lowered that activation energy barrier for the species to favor the same state that the sodium was able to provide or achieve for those oligomers or aggregates. If we look at the collision cross-section area of those species, they're actually different by about 30 angstrom squared, which is a pretty large difference for a small ion. And so then I looked at all of the oligomers that we saw, or aggregates, and here we have the polyalanine calibration right here. And then these are the L-DOPA aggregates. So if we look at the slope or the change in CCS relative to the mass for the polyalanine, it's about a 0.16. However, for the doubly and triply charged L-DOPA aggregates, it's about half to a little less than half of that change, and so indicating a significantly more compact shape than the polyalanine linear polymers. And so this, I then looked at what we showed earlier, the polystyrene sulfonate oligomers, and they were complexed with polylysine, and Mehmet in this paper looked at the change in slopes for the different complexes and showed that the polystyrene sulfonate and polylysine form the smallest or most compact structure of all of the things he tested, and his change in CCS relative to the mass charge was about double what we saw for our aggregates. I didn't really get into the higher mass species because I didn't calibrate past around 2000 with the polyalanine, but it's worth calibrating with the larger protein standards as well to make sure that that trend stays true. So from there, I was then looking into literature about all the different supramolecular models of eumelanin. And so someone in 2015 had done a study that was pretty much the exact same thing that we did, except they were able to show it all with protonated species. They did not do any ion mobility, so they didn't have any dual conformations that were formed, but they did show that these aggregates are forming in solution and not specific, or not ESI induced, but rather native to the potential formation of the eumelanin itself. Here we have the DHI oligomers chelating to different metals and forming these structures in non-covalent ways. And then ultimately, these two structures have been reported recently, or this angstrom separation between the non-covalent aggregates has been a big point of interest for a lot of the community. And someone most recently did both DFT and molecular dynamics studies on the, it's essentially a porphyrin-shaped compound with 4 DHI units. And they looked at that being at a bend or a non-planar conformation because of the way that the oxidation can change that overall conjugation. And from that, they were able to show these really cool large macromolecule structures of up to, I think, 120 oligomers per aggregate. So I'm currently working on modeling these species using DFT calculations, starting at the dimer, and then we're going to build up and try and compare that to our tetramers data that we have. And then from there, also try to do this similar study with molecular dynamics once we've optimized the initial kind of proto-molecule for aggregation. And so here, my colleague, one of the computational chemists here, has done DFT calculations for me, and then I calculated MoCal CCS values, and he gave me about 10 structures and normalized the energies. So these are the 2 lowest energy species, and they exhibited 2 different CCS values on 15 angstrom squared. So here's an example of one of them showing kind of this, like, rather than this pi-pi stacking, it's more of like a C-shaped stacking of the species. And then the larger one was, sorry, no, this one is the larger one from that C shape, and then the smaller one is a more compact pi-pi stacking species as well. So our data for that exact species as the protonated structure showed actually a CCS in 40 angstrom squared. So for me, that suggests that they're actually unfavorably linearly hydrogen bound and then kind of collapsing on themselves into these more stable states as they are provided enough energy. So additional experiments right now that we're working on are just other bottom-up stuff using ESI as a reaction vessel for dopamine and cysteine oxidation using the microdroplet theory, and then studying the products that form from droplets by collecting that spray and then comparing it to bulk solutions using ASAP. That was something that my undergrad had done at the most recent ASMS meeting. And then we're also interested in top-down, and I've done some work on this, actually kind of a lot, using ASAP of these nanoparticles suspended in solution, and then those same nanoparticles treated with fluoride labels to try to see if covalent linkage was happening between the fluorophore and the nanoparticle. I've looked at natural melanin sources through MALDI and ASAP, as well as laser desorption imaging of the coatings that they create. All of this has been inconsistent and irreproducible, suggesting a wide variety of different structures rather than a single polymer forming. It is a very polymorphic and heterogeneous mixture of ions and species. So it's a challenge to do, but it's interesting from an analytical chemistry standpoint. And but basically the summary we have right now is that the L-DOPA building block forms strong noncovalent interactions of up to 6 monomers stabilized by sodium at about a 4 to 6 to 1, about an average of 5 to 1 ratio between the DOPA to sodium. And then the low mass species exist in 2 conformations, these open and closed. And as they increase in size, they favor the more closed species. And so the idea is that L-DOPA might form these proto-aggregates that have been proposed previously in eumelanin nanoparticles. From the solid and solution-based comparison, so far it seems that the solution-based structure ionizes entirely different from the surface-coated and solid-state material, indicating potential cross-linking or different chemistries happening. And then the ASAP analysis of the nanoparticles so far has yielded extremely low ionization efficiency, and I am unable to get the same spectra from the same sample dipped using 3 different capillaries dipped in the same sample. So it's either ionizing things off the capillary very well, or just a very confusing mixture of species that has yet to be understood from a mass spec standpoint. So yeah, to summarize, we talked about the UniDec deconvolution for homopolymers. I talked about it being greener, but as Brian brought up, you guys are much more advanced in your separation techniques than we might have here. So it could be something that's also coupled to GPC and SEC to validate the masses obtained from those techniques. We looked at the selective noncovalent adduct protein probes using polystyrene sulfonate for rapid site-specific analyses of proteins. And then synthetic melanin nanoparticles being just very challenging to analyze and extremely complex. So the challenges seem to be similar with papers I've read about lignins and petrochemicals being all the isobaric and complex isomeric species that are present. And so ultimately, I think that just a diverse mass spec range of methods are required to ultimately gain a full picture of this material. So thank you all for listening to my talk. I know it's kind of a bunch of different things, but hopefully someone found something interesting at one point. Yeah, I'd be happy
Thank you
to
very much,
go
Addie.
back and make some questions too.
Any questions from the attendees? I do have a question myself just about the ESI-MS spectrum of the L-DOPA. Does it change over time? Do you have a different adducts over time? If you infuse at the beginning and then wait for an hour, you have the same adducts being detected, or it's getting more and more complex because you have some oxidation induced by ESI or in solution?
Yeah, it's been tough. At about 4 hours, they start to precipitate out. I did a study with someone in polymer science, and she was just studying how much of she had the dynamic light scattering. And yeah, at about 4 hours, the aggregates or species that are forming reach a particle size that at that point is not suitable for our ESI capillary. But this has been, this is a 30-minute analysis approximately, and then I've done about an hour. But I haven't done much further than that out of fear of just kind of clogging the capillary with potential crustated material. But yeah, it is important to look at how that is changing, definitely.
Okay, thank you.
I have a question because I learned a little bit actually from Addie's talk right now myself. Would that deconvolution method help you giving molecular weight distributions of individual components in mixtures?
Yeah, I think
Let's say
so.
if you have a blend of polymers, because if you do GPC, you get the whole thing.
Right.
I'm trying to find out
Yeah.
now whether there is an advantage based on what Brian Katzenmeier said.
You
I mean,
should
the
be able to see
GPC will
that.
give you the molecular weight distribution of the whole thing. Now, your method could be an advantage if you could get individual molecular weight distributions of components that are not resolved in GPC, and end group functionality distributions, if you can separate that, and so on. So it's
Yeah, and groups, defects, low mass, high mass stuff. You
defects,
should be able
low
to
mass.
pick out
Thank
all
you,
of that and
Tony.
get number average molecular weights. Now, I mean, I have to put a plug in for David Strands there. I use Polymeric to do that.
Oh, we do
But
too.
it doesn't
We have
necessarily do all the
We
stuff you're
do
doing, though.
too,
Yeah.
quite heavily. But I think for even, I mean, in combination with that, I think that would be very useful. I mean, GPC, and the other thing is GPC is dependent on standards. So for most people, it doesn't matter. I mean, whether they use polystyrene standards or not, they report whatever they report. But sometimes if you need more accurate molecular weights, I mean, then your technique would have an advantage. So,
Well, hey,
Yeah.
Chris, speaking of accurate molecular weights, I mean, if you don't want to use RI and standards, you can go
Yes.
to, that would be a relative molecular weight data. You could also go to a absolute molecular weight determination using techniques like light scattering. So
yes,
that would be the other option there if you wanted to go down that
Right.
route. Yeah.
I know.
But using Addie's approach, plus you couple that with MSMS fragmentation, you have the chemical connectivity all in one data
Absolutely.
set. Well, you know, multiple data sets. You have all the information that they need.
Yeah,
Beautiful
you're not going to
approach.
get the molecular information from a size exclusion technique, that's for sure.
No.
That's right, because you have essentially the mass spec detector then instead of the light scattering detector giving you this, what Tony said, this additional structural information, and maybe ion mobility at the same time or whatever the
Goodness,
mass
yeah.
spec has. Yes.
And
Yeah, I was using only a very small fraction of that software. I know he's already set it up for deconvoluting ion mobility and different chromatographic elution times. So there's a lot more to learn and a lot more to do with it too. So exciting potential.
how fast is the convolution? Like, would it be possible to imagine having this in real time? Like you're querying data, and like after
Ooh.
a second, the computer is giving you both the actual mass spec and the decomposed mass spec, so at a glance in real time.
You would definitely need to be pretty machine learning savvy. I think at that point you have to extract the text file, or I guess I haven't figured out how to do it, but I know that they're capable. There's ways that you can upload waters, thermo, and adjuvant, I think, data directly without converting it to the text file. So it'll read that whole raw data set from MassLinks or various softwares. So you could potentially have it programmed to read your live data. That would be pretty cool, definitely.
Yeah, like
I don't know how to do
if
that.
you can convince Waters or I don't know, JOL or Broker to add this to their acquisition program, you will have the real time, real data in the
That would be
like
really cool.
uncharged mass spec. Yeah. Okay. Okay. Any other questions from anybody or any other topics you want to discuss today?
Yeah,
Yes, if I may go back to your second part regarding the measurement of the distance. It's cool work, but I'm wondering, the principle is based on the assumption that activation of the complex does not modify the conformation of the whole complex before it fragments, right?
Yes, and that's definitely something I want to look into with proteins that then have a bound active site, and seeing if that, like if tagging it with the polystyrene sulfonate induces some sort of allosteric change that then in solution we can no longer say that the adducts are forming. But there's definitely conformational things that we're assuming right now, and that's why we're doing it all on known proteins and then validating it against their PDB files in the protein data bank, just to make sure that we are validating the results we get to other techniques as well.
Okay, thank you.
I had a question on the last part. Is the work you did with ASAP you said was really sort of random. You were getting different results for different. I was curious because you mentioned that a lot of the work in the past is done using pyrolysis GC or pyrolysis GCMS. Do you get a sense that their data was reproducible and they got kind of the same answers type of thing?
I think so for the most part. I think people have a good understanding of the monomeric units or backbones of the material. I think it's just how they're connected that's been challenging. And so at that point, because of the diversity of the different monomers, it's hard to say whether some are forming from the pyrolysis, whether that's a connective covalent monomer that is formed naturally. So I think a lot of it's mainly just been elemental and monomeric analysis from that standpoint. So it's
Yeah, I
hard
was
to
thinking of it because the ASAP and pyrolysis are sort of relatives of each other in terms of a thermal decomposition pathway that happens to give you data. I was curious as to why maybe you just need to put a lot more energy into it thermally to get reproducible data.
Yeah, definitely. So the natural extracts that we've done it on are also challenging because they are complex with lipids and other biomolecules that might ultimately covalently link to melanin, has been shown in fungal melanin and stuff. And so it's hard to then remove that picture from the analysis in terms of the top down. But with the synthetic material, it's also challenging because people have proposed that the solution-based aggregates might then covalently cross-link once they've been surface modified or something. So it's hard to say if that's happening on the capillary or in the source too. But there's a lot of controls that need to be considered for this.
Yeah, it's definitely a challenge.